for phenotypic changes, that is proliferation, apoptosis, or something. Now, the goal of the PhD project was to investigate the roles of input signals on the final response in T cell signaling, the roles of different signaling molecules involved in T cell signaling, the roles of protein-protein interaction strength, that is rate of reaction or kinetic parameter, and how the cellular responses, that means fully activated ERK, is controlled when there is a change in the concentration of signaling molecules, or change in the rate of reactions, or change in the interaction nature, for example, the loops, feed-forward loops, feedback loops, or crosstalk between the pathways. The goals which I have achieved in my PhD thesis work, so there was basically two work which was published as a goal. One was published in PLOS One, that is, as mentioned here, where we have basically investigated the rate of reactions. And second one, which was associated with effect of changes in the concentration levels of signaling molecules. Third one was crosstalks of signaling pathways. What I did basically, second and third goal, I merged into one paper, and that was published in scientific reports. So next part is here, first goal, which was published. So basically, before going to introduce the first goal, I'm just introducing how a signaling network could look like as a network. So the pathway looks like a straightforward, but in general, it does not happen to be in that way. So here, for example, the green S node, there are two things here. You can see the node circles and the links, that is the nature of connectivity between the different proteins. And then here, the nodes are of different types, like input signal, inactive nodes, partially active nodes, fully active nodes, and the complexes formed as a result of interactions, as a result of chemical reactions within the cell. Now, how should we consider what is the, like, for a layman to understand how the protein-protein interaction, like, strength is changing? Here we are just saying the rate of reaction or kinetic parameter, for example, KD. In the first reaction, you could see 1.0 and it is changed to 3.0. That is the mutation in the rate of reaction. Now, addition of new proteins in the minimal model. So this is also one kind of thing which the people needs to know, how to add the new proteins or new nodes in a small model, and how the mathematical models are working. So mathematical model here, we have used evolutionary algorithm to simulate this entire model. And in evolutionary algorithm, it is just to create the networks, then calculate the dynamics of each protein. That means the overall dynamics of the network, then evaluate the fitness of the network, then go for elite selection. That means top 25% or top 30% of the best networks are selected based on the fitness. So in order to consider the network as a good network or bad network, we calculate some kind of fitness values for each network. Then go for selection and mutation. So mutation could be in rate of reaction, concentration, or addition or removal of the existing nodes, or addition of new nodes. Then create the new copies of the selected networks. In order to maintain the total number of networks must be equal to the population of the network, or population or the network which we created before we start the simulation. And this is what we are repeating again and again for 200 times or 300 times. Each repetition is called as generation. So now we are, and this work has been published in PLOS One in 2012. Now, I'm just going one by one. Role of interaction strength, how the fitness values are changing when the rate of reaction is less than 10, when the rate of reaction is less than 30, when the rate of reaction is higher than 30. So that means here we are also K less than 10. That means the rate of reaction is very, very weak or small. Then we are increasing the rate of reaction, how the networks are behaving. Here, what we are seeing, for the rate of reaction less than 10, the fitness is not reaching to the maximum level. That means all the networks are not going to be considered as the fit network. But when we are increasing up to certain threshold, then all the networks are basically trying to reach to the best fitness values. And here we could see easily in the networks how the responses are behaving. So this one, how the each network is behaving in different generation. In the beginning, G1 means generation 1, G50 means generation 50, G150 means generation 150 or iteration number 150. In each generation, we mutate the rate of reaction. One rate of reaction we select randomly and then mutate it. So what we are observing that with the passage of time, the network is achieving better and better response, and these responses are called as sustained responses. So that means once it is activated, it remains always active. Now we are going to check another thing, how these networks are. So this was basically evolution of kinetic parameters, how these kinetic parameters are evolved in each generation. So what we are seeing, so mostly the rate of reactions, all the reactions are trying to achieve the higher values. That means they are trying to go for maximum values. And here also in the lowest kinetic parameter